Hello, and welcome to my video on mRNA point mutations. So here we're given a DNA sequence, and we're supposed to find a chain that's going to give us 4 amino acids. So since we know that transcription goes from the 3' to 5' direction in relevance to the DNA, transcription is either going to go from right to left on the top strand or from left to right on the bottom strand. So let's use the top strand as our template first. Transcription is going to occur, giving us this RNA, and then translation is going to occur, and we find our reading frame. There's our start codon. Here it is, and there's our stop codon, and as you can see, we are given a protein that is 4 amino acids. Now we're going to take this and we're going to mutate it a bit. We're going to go through these 4 mutations: the missense, the silent, the nonsense, and the read-through. I'm also going to talk about whether or not that the mutation is a transition or transversion, depending on whether it's a pyrimidine or a purine. So with the missense, one nucleotide is going to change and it's going to alter only a part of the protein, which could change the function. I kind of see that as like the different there, and there words. They have different functions, but they're spelled differently. So we're going to change this uracil into a cytosine. Now this is a transition because they're both pyrimidines. Here are the 2 reading frames. And as you can see, the second protein is changed, giving us a new protein sequence. And then here's the coding strand. For the silent, I kind of see that as like words being misspelled. There's a part that is mutated, but it doesn't actually change the protein. You still have the same function. So we're going to change this cytosine into a guanine. Now this is a transversion because we're going from a pyrimidine to a purine. Here are our 2 reading frames, and as you can see, they give the same protein. So our silent protein is the same as the wild type. And here is our coding strand. Now with the nonsense, we are given a mutation where the codon is changed to a stop codon early. And I kind of see this as like stopping during construction work because you're not expecting it. So we're going to change this adenine into a uracil. Now this is a transversion because we're going from purine to pyrimidine. Here are our 2 reading frames. And as you can see, our nonsense mutation has given us a shorter protein strand with only 3 instead of 4. And then here's the coding strand. For the read-through, we are given a mutation at the stop codon, and it changes it so the protein doesn't stop producing amino acids. So I kind of see this as like a broken traffic light. We were supposed to stop, but you actually just keep going. So we're going to change this adenine to a cytosine. This is a transversion. And as you can see, our second reading frame is much longer, giving us at least 3 more proteins. And there's no stop codon, so it's technically still going. And here's our coding strand. In conclusion, here are our products. You can see how the sizes change, and you can also see how the coding strand barely changes at all. There's only 1 mutation, but it can still give us so many different changes.